Okay, we're looking at a topic on plant structure. This is the syllabus objectives, and as we go through the slides, I'll put the relevant bit of the syllabus that we're looking at. So now looking at plant structure in general. What are the roots for? The roots anchor the plant in the ground. The roots also absorb water and minerals. The water enters through root hairs, and minerals such as nitrogen enters through the roots as well. The stems are there to support the leaves and flowers, to support the aerial parts of the plant. Stems have vascular tissue for transporting water upwards and food away from the leaves. Some stems can be modified for food storage. If we're looking at the structure of the stem, where the leaf grows is called a node. So in the diagram, a node is the attachment point from the leaf. And often just above the node is where we're going to have a bud, an area of new growth called an axillary bud. And this can grow into a new branch. So in this part, so we have the new branches growing there. It's growing from what's called an axillary bud, which is just above a node where a leaf joined the stem. So what do leaves do? Leaves are the organs of photosynthesis. Leaves photosynthesize. They use the sun's energy. They absorb water from the that's taken up through the roots, and they take in carbon dioxide from the air and they use it to make food. So the leaf is the organ of photosynthesis. The flower is an organ of reproduction. The flower has the male and female structures, and it produces seeds enclosed in the fruit for reproduction. Now, in this, we're looking at the word meristem. What does meristem mean, and where would you find it? Meristem tissue is made up of cells that are rapidly dividing, cells that are multiplying, producing new cells. Meristem in a plant is found at the shoot tip, the very, very top of the tip. It's also found in the root tip. So they're the main areas where we'd find meristem. We'd also find meristem maybe at the area of the side branches, and we'd have lateral meristems just underneath the outer epidermis to allow sideways growth or growth in width. But the main meristem that I want you to concentrate on are the shoot tip, called the apical meristem, and the root tip down here called, again, it's another apical meristem. What does meristem become? Meristem will divide to form new cells, and those new cells will develop into one of three main types of plant tissue: dermal, ground, or vascular. Okay, so we have three main types of plant tissue, three main groupings of cells when it's inside plants. Dermal tissue is found on the outside of the plant. It protects the plant. Root hairs, for example, in the roots are modified dermal tissues that specialize in absorbing water. Dermal tissue on the stem is there to protect the plant against water loss and against fungal infection. Okay, so we have the dermal tissue on the outside. Ground tissue provides support for the plant and makes and stores food. So a lot of the cells within a plant, they're referred to simply as ground tissue, for storing food or for making food. Vascular tissue, these are the veins that you'll find inside the plant. They transport water upwards from the roots, and they also transport food away from the leaves. So in a slice through a stem, called a transverse section through a stem, we can see the veins. The veins are called vascular tissue. Here's another diagram of vascular tissue. And if we examine that, we'll see that the outer part of the vascular tissue are made up of phloem cells, which carry food away from the leaves. And the inner part is made up of xylem cells, which carry water up from the roots. These cells in here are known as ground tissue. The outer ground tissue is often referred to as cortex. The inner ground tissue in this diagram is referred to as pith. But for your course, what you need to know are the three types of tissue: vascular, dermal, and ground. And we'll meet phloem and xylem shortly. So vascular tissue transports substances within the plant. Now, as in the syllabus, we should also be able to recognize a longitudinal section. This is a lengthways slice, so looking at the stem from the side. Okay, from the outside, you have the dermal tissue, and then you have some ground tissue, And the yellow is the phloem. The phloem is carrying food away from the leaves. And the green here is the xylem. The xylem is carrying water upwards from the roots. Now, we're looking at what a cross-section through a stem looks like. So the location of the tree tissue type in longitudinal sections of the root and stem. So now we're looking at the root. So slice across the root, you'll notice that the xylem is a star-shaped collection of cells at the very center, and the phloem are smaller clusters within the arms of the star. So this diagram is from a past exam paper. It's the kind of one you should recognize. So the other labels on this, we have, how do we know it's part of a root? Because you can see root hairs. Okay, and we have this characteristic star-shaped xylem at the center. Now a lengthways slice through a root. Okay, so just the root tip is where we have the meristematic cells. So these are the cells down here that are rapidly dividing. The root tip is covered by a very tough material called the root cap because the root has to push its way down through the soil. Just above the meristematic zone, this area here, we have the zone of elongation. In this part of the root, the cells are simply stretching. They're gaining, expanding due to water, and they're gaining full size. The zone of elongation. As we go up a bit higher, we have the zone of differentiation. This area here, newly formed cells are developing either into dermal or vascular or ground, and some of the dermal tissues are developing into specialized root hairs. So in this part of the root, which is just above the tip, cells are beginning to differentiate and get different jobs. Okay, so what's important in this diagram? Meristematic cells at the tip. Behind that, newly formed cells are stretching. And behind that again, newly formed cells are now developing into different types. They're differentiating into either ground or vascular or dermal. Now we're going to look now at xylem cells in a bit more detail. Xylem cells, these are the cells that carry water. They're dead at maturity, so once they reach full size, the cell contents die away and all you're left with is an empty cylinder. So they often form long continuous tubes like drinking straws, and to strengthen them, they're covered with spiral bands of a very tough material called lignin. Okay, so we have spiral bands of lignin around the outside. They prevent the xylem vessels from collapsing. They also have holes. These holes allow the lateral or the sideways movement of water. These holes are called pits. So this is from a Leaving Cert exam. Okay, how do you recognize it as being xylem? Number one, it has pits, and number two, it has lignin, spiral bands of lignin. Okay, this particular xylem vessel would form a long continuous tube, so one cell stacked on top of another cell and so on, forming a long empty tube. Other types of xylem cells are called xylem tracheates. They have lignin as well, they have pits as well, but they have tapered ends. So these tend to overlap each other rather than stack one on top of the other. So here are two types of xylem cells, xylem vessels and xylem tracheates. Now to look at phloem. Phloem are living cells that transport food away from the leaves. The food is transported in a soluble form called sucrose. So phloem cells don't have lignin. Okay, they're living cells. But how do we recognize them? They have a characteristic plate that separates one cell from the next called a sieve plate. The sieve plate has little holes punched in it. It allows the cytoplasm to move and flow from one cell into the next. This is how the food, the sucrose, is transported. So the cytoplasm and the sucrose moves from one cell to the next through the sieve plates. There are also companion cells found beside these sieve tubes that seem to control the activity of the sieve tube in some way. So there are two types of phloem cells: phloem sieve tubes and phloem companion cells. Companion cells don't have any obvious function to us. All we have to know is they have some control over the sieve tube. The sieve tube have this characteristic sieve plate. Now, this is also necessary to look at in this chapter, identification of dicot and monocots under these headings. Okay, so we can classify plants, we can group plants as being either monocotyledon or dicotyledon. A cotyledon is a food store. Some plants have one food store in their seeds, and some plants have two food stores in their seeds. So a monocotyledon plant has one food store in its seed, one cotyledon in its seed. Okay, now other differences. Monocots such as grasses are often soft-stemmed or herbaceous. The stems of dicot trees such as oak become woody as their older tissues lignify. Okay, so dicot plants can become larger and wider and more woody. Monocots have flower parts in trees and sixes. Dicots have flower parts in fours or fives. So if you were to count the number of petals, okay, there's 3 petals in that plant. Okay, it's a monocot. There's 5 petals in this flower. It's a dicot. If we look at the vascular bundles, okay, as we would have drawn already, the vascular bundles in a dicot are in a neat little ring close to the dermal tissue on the outside, whereas in a monocot, the vascular bundles are scattered. Okay, so that's a big difference. Scattered vascular bundles in a monocot, but vascular bundles in a ring in a dicot. Okay, as I've said, monocots have one cotyledon food store in their seeds. So this is the seeds here, one cotyledon, that some corn, as in sweet corn, one cotyledon food store, and there are 2 cotyledon food stores in this seed. Okay, and when we do a chapter on plant reproduction, we'll go into what a cotyledon is in more detail. Many monocotyledon plants have parallel venation in their leaves. Okay, so in this plant here, if you look at the veins in the leaf, they're all parallel to each other. That's a characteristic of a monocot. The veins in this plant here, they're branching and netting. Okay, that's an example of a dicot. So looking back over the syllabus, woody, herbaceous. Okay, woody is a dicot, herbaceous is monocot. Arrangement of floral parts in trees would be a monocot, in fours and fives would be a dicot. Vascular bundles, okay, scattered vascular bundles is a monocot. Vascular bundles in a ring is a dicot. Cotyledon, okay, a cotyledon is a food store inside the seed, and it can also develop into a leaf. The cotyledon can develop into a leaf. So if there are 2 seed leaves, it's a dicot plant. If there's 1, it's a monocot plant. Now, just to look at how plants can store food. Plants can't grow that well over the winter because there isn't enough sunshine for photosynthesis to happen. So if a plant needs to survive over winter, it needs to have a store of food to allow it to start to grow again in the spring. So plants have structures modified for food storage. Okay, we have to know one example from a root stem and a leaf. So the modified root, a good example of that is the carrot. The carrots produce a long taproot, okay, for storing food. So photosynthesis happens throughout the summer and the food is transported down to the root. The cells in the root swell with stored food. And this stored food provides the energy for growth the following year. Likewise with a potato, okay, the potato is actually an underground stem. Okay, you would think it's a root because it's underground, but it's actually a stem. Okay, known as a stem tuber. So they swell with food. Food is made in the leaves by photosynthesis, is transported down through phloem to the underground portion of the stem, and they swell with the stored food. So the potato is an example of a modified stem called a stem tuber. It also acts as an organ of reproduction because the following year, each of these potatoes can develop into a new plant. And a bulb is an example of a modified collection of leaves. So it has a modified or a specialized bud at the center, but the leaves that surround the bud are storing food. So an onion, for example, all of those layers in an onion are called leaves and they store food. So I've highlighted there, carrots store food in their roots, potatoes store food in a specialized stem called a stem tuber, and an onion stores food in specialized leaves in a bulb. Now, in this chapter, there's a mandatory activity where we have to take a slice, a transfer section through a stem, a die-cut stem. Okay, so here's a summary of the process. Cut a thin slice of a section of a busy leafy stem. So it's important that we name the plant we're going to use. We're going to use a scalpel or a backed blade or a safety blade to do this. You can hold the stem in your hand and cut the stem at right angles with the blade. Place the section into water to allow the cells to recover, to allow your section to recover, to absorb water, to remain turgid. Okay, then you can repeat that a number of times so you have a number of sections resting in water. You can select the best section and the thinnest section using a paintbrush and transfer it to a microscope slide. The paintbrush has delicate bristles on it that won't damage the section. So on the microscope slide, then add a drop of water and add a cover slip, one edge first. Okay, gently lowering it down like a trapdoor to avoid trapping air bubbles. The air bubbles just make it more difficult to see your specimen. The purpose of the cover slip is to prevent the specimen drying out. So examining under a microscope, place the microscope slide onto the stage of the microscope. So it's being specific with the part of the microscope, you're putting it onto the stage of the microscope, turning on the light source, starting with low power the objective lens, and look through the eyepiece. Okay, so the eyepiece will have a magnification of 10, and you combine that with the low power objective lens, or the in our case, the medium power objective lens would have a magnification of 10. So 10 by 10 will give you a total magnification of 100. Okay, so I'm just referring back up to the title, the syllabus title here. We're supposed to examine under 100 and 400 magnification. Adjust the coarse focus knob and the fine focus knob to view the section clearly. All of these statements are what the examiners would expect you to include in your description.